Hello everyone, good morning, and thank you so much for being part of my talk today. I'm going to be talking a little bit about my postdoctoral research and the work we're doing to identify the inflammatory mediators, the cause behind the inflammation that we see in the brain, and the sex differences in Alzheimer's disease. So I thought I'll start with a little bit about me. I hail from a little town in the southern part of India called Chennai. I did my undergrad in biotechnology. I moved on to New York then to continue my research in immunology at the State University, New York of Buffalo, and I did my master's in microbiology and immunology. Moving on from there, I went to do my Ph.D. at the University of Rochester Medical Center. At the university, I got to do a number of different projects related to immunology. And these were related to HIV, cancer immunology, fungal pathogens, multiple sclerosis, and dementia. So through these projects, I developed my most important interests in neuroinflammation, innate immunity, and autoimmunity. So given my trifecta of interests, I looked around for a suitable postdoc, a postdoctoral fellowship, and that's when I hit upon Dr. Sonia Sharma, my current boss at the Jolla Institute for Immunology. She asked me if I would be interested in looking at what the mediators of Alzheimer's disease could look like. She asked me if I could take what I've learned in my dementia models and apply it in her human cohort to identify these mediators. So just to give you an overview of Alzheimer's disease, the next few slides are going to dive deep into this disease and learn what we know about this disease today. So Alzheimer's disease is a global burden of dementia, and what we see here in the prevalence rate is that this is a global disease. There are many countries that are affected by this disease, specifically the United States, where you can see the color is much darker, which indicates a higher prevalence rate of this disease. What's more alarming, though, is that when you look at the past 10 years and look at the growing rate, or what's known as the incidence rate of these diseases, it seems to be increasing as well. Interestingly, technology has grown leaps and bounds. We've been able to discover medication for so many different diseases, and yet Alzheimer's disease is still on the map and still growing. One of the reasons identified as a contributor to this developing rate or growing rate is the socio-demographic index. What this study in 2022 identified that when you were to consider the socio-demographic index, which indicates how rich or poor the country is, you observe that there's a direct correlation between the incidence rate and the socio-demographic index, as seen here. What this tells us is two things. One, in countries that are richer, in countries that are higher up on the socio-demographic index, have a better healthcare system to identify patients with Alzheimer's disease. This is great. But another factor, a contributing factor, is that patients living in these societies live longer, and therefore you see more of the age-related or aging-related diseases such as Alzheimer's. Now, when you look at the types of dementia, there are several different types. Alzheimer's disease forms the biggest part of this pie, but you also have other diseases such as vascular dementia, frontotemporal dementia, dementia with Lewy bodies, etc. It's very hard to specifically identify which type of dementia you have. There are some biomarkers, but the most likely event is that some patients could be developing more than one kind of dementia or disease. What's shocking is that even amidst COVID-19 related deaths, Alzheimer's disease still makes the top 10. So let's talk a little bit about the hallmarks of the disease. Dementia is the main hallmark of the disease. So what is dementia? It starts with problems in communication, changes in mood or behavior, poor judgment, and it can lead to even forgetfulness of day-to-day activities or function. In extreme cases, it could lead to withdrawal from work or social activities, and eventually you might need aid or help to do your daily activities. Over a period of time, the patient loses their cognitive abilities, and eventually this could also prove to be fatal. So another hallmark of this disease is the brain images. So in a healthy brain, you see that the brain is a healthy size and the neurons look healthy. There are no clumps of proteins inhibiting the neurons from functioning. But in an Alzheimer's brain, what we see through imaging techniques is that they develop protein clumps both inside of the neuron, called the neurofibrillary tangles, or NFTs, and outside of the neurons, called amyloid beta plaques. So these proteins exist in our body primarily to function. They have a physiological function. But under certain conditions and under inflammatory conditions, they tend to misfold and clump. Now, when you don't take the garbage out, you block the streets. And that's pretty much what happens in the brain. This blocks the communication between the neurons, eventually leading to the death of the neurons. And what you see is that, unlike the healthy brain, the brain begins to shrink. So now I'm going to walk you through what the disease timeline, the brain changes, and the tools and the therapies that we have to study this disease. What's interesting about Alzheimer's disease is that we tend to classify patients as clinically diagnosed Alzheimer's when they start to express certain neurocognitive deficits. But the reality of the situation is that the disease starts possibly up to 20 years ahead of you showing the symptoms. So these phases are known as preclinical and prodromal phases. At the preclinical stage, we have no symptoms. There's very few biomarkers in the blood to show or detect the preclinical stage. And as you progress through these stages, you tend to have more of those clumps in the brain, which can be picked up in the fluids, such as your cerebral spinal fluid and your blood and plasma, etc. You have increased neuroinflammation, neuronal loss, systemic dysfunction of metabolism, and brain shrinkage. Now, the diagnostic tools for the past couple of decades have remained gold standards. These are the brain imaging techniques, such as MRI, CT scans, and the fluid tests, which pick up these misfolded proteins in plasma. Now, unless you show symptoms or neurological symptoms, you're not classified as having clinical AD. And this can be a real problem. When you think about what the current therapies show us, most of the therapies begin when you start to show symptoms. But as I've just explained here, the actual disease itself begins way ahead of that. It's no wonder these late interventions have been non-disease modifying. They treat symptoms, which are the amyloid beta plaques and the NFTs, and most of these drugs have failed clinical trials. But what we are working on right now and for the future is to identify those markers, is to identify early intervention and disease-modifying drugs that can reduce the inflammation in the brain. So what are the different types of AD? We've heard of the familial AD, where it could run in the family. This makes up only about 10 to 15% of the cases that the doctors see. The early onset familial forms of AD have an autosomal dominant inheritance linked to 3 genes, APP, PSEN1, and PSEN2. But most of the cases that you read about are sporadic AD. They're late onset, unlike the familial AD. There's one known genetic component, which is the ApoE protein and the variants of ApoE protein, which usually function to keep your brain working. But under certain circumstances, certain variations of this protein can be associated with increased cases. Apart from the genetic component, you also have a number of other risk factors. For example, blood cholesterol, hypertension, vascular injury, and another not mentioned here is smoking, and of course, the female sex. Regardless of how AD develops, the hallmark features are seen in both, which leads to the dysfunction of neurons and eventually cognitive impairment and death. What's strikingly unique about Alzheimer's disease is the strong female bias in AD. 1 in 6 women versus 1 in 11 men are at a risk of developing dementia. Autopsy reveals more plaques and NFTs in women. Greater brain shrinkage in women is accompanied by rapid decline of memory and cognitive function. Factors include aging, sex hormones, and molecular causes are unknown. That is the current challenge in understanding the molecular basis of this disease. So the current gaps in research are that there are few to no disease-altering medications for AD in decades, but also that research on sex-biased AD is equivocal and requires further investigation and understanding of the molecular and cellular factors that drive this disease. The biggest challenge in studying immune system diseases are that diseases of the immune system affect men and women differently. There are profound sex-specific differences in the biology of men and women in the immune system. Many immune diseases and conditions affect women and men differently. I'm going to walk you through a few diseases where this profound differences is seen. Number 1 on my list is COVID. Men have a 10 to 20% higher rate globally when compared to women. On the flip side, women are 20% more likely to experience long COVID, a condition where even after you've cleared the virus, you tend to have systemic inflammation, and this recent research shows that this could also cause neuroinflammation. Another disease, as these images probably have already given you a clue, is the heart attack and cardiovascular diseases. If you look at these images here, these are images from a Google search for cardiovascular diseases and heart attacks. You can see that there's 1, maybe 2 female images there, which shows you that women don't experience heart attack the same way men do. Number 1 cause of death for both men and women, yet differences in how the cardiovascular disease manifests in men and women are high. Before menopause, women have a lower CVD risk than men. After menopause, the risk is reversed. This shows that aging plays a role in the inflammation that we pick up in diseases. Different symptoms, such as chest pain or angina, swelling, signaling heart attack, often lacks in women. They are almost different diseases at this point. So what I've shown you here is that while women are prone to inflammatory diseases and autoimmune diseases and can raise a really high, robust immune response, men, on the other hand, lack a robust immune response to pathogenic infections, making us really very different in how we react to diseases. Autoimmune diseases disproportionately affect women. And this is just a handful of diseases listed here that are autoimmune diseases that affect women disproportionately. Why am I talking about autoimmune diseases? Because what we've come to appreciate in the last couple of years is that Alzheimer's disease primarily is an autoinflammatory disease of the brain, as seen here with the Alzheimer's brain, in comparison to even the healthy elderly brain, has much more inflammation. So really, understanding sex-specific differences on a molecular level can uncover new disease origins, lead to better prevention, deliver personalized treatments. So like I mentioned, the current therapies lack what we at our lab are looking into. What we focus at the Sharma lab is identifying those early intervention molecules, identifying disease-modifying medication, and reducing inflammation. But before we talk about that, let me quickly give you a review of what the immune system looks like. The immune system comprises of many different layers. You have the barriers, which are the physical wall between you and the environment, and this includes the skin, the mucus in your nose and lungs, the low pH of the stomach, and even the tears and saliva. The innate immunity are the foot soldiers. They are the first to respond to an invading pathogen or an infection. And when they attack these pathogens, they send out signals to bring in the cavalry. The cavalry are these strong arms that produce the humoral response or antibody response and kill the invading pathogens. Now together, the right inflammatory signals, these different arms of the immune system can interlock and coordinate the process. One such inflammatory signal are circulating metabolites. Circulating metabolites are substances made or used when the body breaks down food, drugs, or chemicals, or its own tissue. So what do I mean by that? We all know about the central dogma of life, where the DNA converts to RNA and produces protein. But in the process, they produce metabolites that have several functions to play to keep your body working. They have energetic functions and signaling functions that keep your organs functioning. But what we've come to appreciate in the last couple of decades is that these metabolites can now also be part of disease pathogenesis. They could be causing diseases in some cases. So to study these metabolites in our human patients, we decided to use 3 different cohorts. These different cohorts are from different parts of the world. We have the arid cohort, which is from the central or eastern USA, the FINRIS cohort from Finland, and the Alzheimer's cohort from the University of Washington. Now, as you can see in the breakup, in the top 2 cohorts that we've worked with so far, we've already started to see a strong female bias in our dementia patients. What we do is sample the blood sample from these patients over a period of 10 to 15 years. We observe these patients in their preclinical stage, and then identify, using biostatistical methods and metabolomic techniques, the actual metabolites that correlate with patients developing the disease. Then this is where my work comes in our lab at LJI, where we take these identified metabolites and study them to see whether they are pathogenic or not. So before I go into the data a little bit, I wanted to share with you what the hazard ratio is. A hazard ratio measures the effect of metabolite level on developing the disease. So what do I mean by that? On a graph that looks very similar to this, if the metabolite has a number of 1, it was not associated with the disease at all. If it has a number higher than 1, then this particular metabolite increased over time in the blood of patients who developed the disease. So we call them pathogenic metabolites. But if that metabolite has a number less than 1, we call them protective metabolites. But as you can have guessed, these metabolites increase in patients who did not develop the disease. And we believe by knowing these two different molecules, we'll be able to identify protective supplements and prospective biomarkers. What was interesting is that from our first cohort, we only identified pathogenic molecules in our male-specific metabolites. So all those lines there that you can see in blue were these metabolites that were specifically identified in men, and they were all seen to be pathogenic. One such pathogenic metabolite that I'm currently studying in the lab right now is known as the inflammatory leukotriene molecule. Now, research tells us that these leukotriene molecules associate with many different diseases. They've been identified in asthma, cardiovascular disease, and even cancer. So we propose this question: are higher leukotriene levels in blood a good biomarker or biomodulator of neuroinflammation in Alzheimer's disease? To study this, we decided to look at the immune system of the brain. The brain is composed of many different cells, but the most important immune cell in the brain is called the microglia. What we do in the lab is grow out these microglia in a dish, add the leukotriene to them, and see what that does to my microglia. We observed that these leukotrienes inflamed and caused the microglia to die, showing that these are highly inflammatory molecules, possibly biomarkers of neuroinflammation in human beings. Now, when we look at the female-specific metabolites, that's when things get very interesting. We identify both pathogenic and protective molecules. So I'm going to talk a little bit about the protective metabolites here that were found in higher concentrations in younger females. Unsurprisingly, one of the protective molecules that came up was the omega-3 fatty acid. It's an essential fatty acid, which means you need this particular fatty acid for your body to function regularly. It's found naturally in oils of fish, hemp, and blackcurrant. Omega-3 polyunsaturated fatty acids affect brain homeostasis and development. This is already known. Dietary supplements for humans and pets are currently being manufactured. Fish oil is a billion-dollar industry. However, the specific protective metabolites in fish oil are not precisely known. In order for us to study this metabolite and identify its mechanism, I can't experiment on human beings. So that's why we develop mouse models in our lab. What I'm going to show you next is an image of the mouse brain that we can take at the La Jolla Institute for Immunology to study inflammation in the brain and how the fatty acid supplements that we provide these mice can inhibit the inflammation in the brain. The blue cells are the microglia. The pink shows me the specific microglia that we're studying. And the green dots then identify the activated or inflamed microglia. So I'm going to show that one more time. Blue marks the nucleus of cells in the brain. Pink marks the microglia. And the green dots identify those angry, inflamed microglia. And what's beautiful is that we can map out these tentacles to see what's changed in the brain when I feed them the fatty acid supplement. So at present, I'm developing this mouse model at the Jolla Institute for Immunology. We look at a specific region of the brain that I'm about to show you called the hippocampus. The hippocampus is a seat of consciousness in many ways and is a center for thought and cognition. And it's one of the earliest regions that start to deteriorate as you start to develop Alzheimer's disease. What's beautiful about the Jolla Institute for Immunology, and as I've learned over the couple of years, is our ability to talk to donors and to the layman population who really love working with us to solve diseases like Alzheimer's disease. We were able to get a software which could provide much more clearer images of the microglia, which helped me look for small, minute changes that we couldn't earlier. This software was donated by one of our donors at LJI. As you can see, the picture on the left was a fuzzy image of the microglia. I couldn't appreciate the size or the tentacle or the structures that come out of the cell. But with this software, we could perform better analysis, identify individual microglia, and perform studies that can tell me about the number of microglia, the structure, the activation cycle, all of it. So in conclusion, what comes next is the Metabolomics study that can link inflammatory molecules to diseases like cancer and Alzheimer's disease. Metabolite disease association can be pathogenic biomarkers or protective in treatments. We are developing new in vitro and in vivo techniques to study brain inflammation through the activation of microglia. Validation of Alzheimer's disease-associated metabolites is required, as I've mentioned, for therapeutic development. So with that, I'd like to thank my lab, both the present and past lab members, the imaging core at LJI, our collaborators across California, Dr. Mohit Jain at UCSD, now Sapient, Dr. Susan Cheng at Cedars-Sinai, and the Specialized Center for Research Excellence, or the SCORE, principal investigators and committee members who are a big part of the study, guiding me through and navigating these grant applications. And of course, NIH as my funding source. Thank you so much.